Let's learn about cell wall. It is present in plant cell, fungi, bacteria, but absent in animal cell. Cell wall is a tough, rigid layer that surrounds the cell membrane to provide structural support and protection. Plant cell wall contains cellulose. That means cell wall is made up of cellulose in plant cell. You know that when we place a cell in hypertonic solution, the cell will lose water and become shrunken. Like that, here the cell loses water through osmosis. This results in the shrinkage or the contraction of the cell membrane away from the cell wall. This phenomena is called plasmolysis. During this time, the cytoplasma will come away from the cell wall. Actually, cell is living and the outer covering of the plant cell wall is non-living. It is because the cell wall is made up of cellulose that makes the cell wall non-living. And fungal cell wall is made up of chitin. And bacterial cell wall is made up of peptidoglycan. So, cell wall is the outer layer in these cells, and plasma membrane or cell membrane is the second layer in these plant cell, fungal cell and bacterial cell. Can you imagine why does an animal cell do not have a cell wall? Let us understand why the plant cell have a cell wall. Consider this comfortable place for animals and plants. Suddenly the weather has changed. Rain, thunder, lightning. At such times, animals can usually find a shelter to hide, but not plants. They cannot move to find a shelter. They have to stay where they are. In this situation, a delicate cell membrane won't give plants enough protection. That's why plant cells have one more protection, cell wall. Animals can easily move from one place to another. So animals do not require a cell wall. Plasmolysis is the process of shrinkage or contraction of the protoplasm of the plant cell as a result of the loss of water from the cell through osmosis. As we already learned that when we place a plant cell or animal cell in a hypertonic solution, the cell will lose water and the cell will shrink. Normally, people use royo leaf for experiment because it contains colored plastids which can be clearly visible. Take two glass slides, take a royo leaf from the petri dish, fold the leaf, tear it along the lower side of the leaf. Using a forceps, pull out two small segments of thin transparent layer. Place the layer on both the slides. Using a dropper, add 1 or 2 drops of sugar or salt solution on the layer. Observe the slide under a microscope. We will observe cell wall, cell membrane, cytoplasm, nucleus and small green granules called chloroplast. The stomata cells are also visible. Now, on the Second slide, add hypertonic solution of sugar or salt. After a minute, observe it under a microscope. We will observe shrinkage in the cell content due to plasmolysis. Similarly, prepare a slide of a boiled leaf under a microscope. We will find discolored stomata and other organelles. Now on the same slide, add a drop of hypertonic solution. Observe it under the microscope after a minute. You will find that plasmolysis did not appear because boiling kills the cells in the leaf. What do we conclude from this activity? Osmosis takes place only in the living cell, not in the dead cell. Cell wall permits the cells of plants, fungi and bacteria to withstand very dilute hypotonic solution without bursting. In such media, the cells will gain water by osmosis. The cell swells, building up pressure against the cell wall. At the same time, the wall exerts an equal pressure against the swollen cell. Here, the cell wall acts as a pressure vessel, preventing overexpansion when water enters the cell. So, cell will not burst. Cell wall provides rigidity and shape to the cell. Cell wall is freely permeable. It means cell wall will allow free movement of molecules in plant cell. Free movement of molecules like water, gases will easily move across this cell wall. Lastly, the cell wall provides protection during unfavorable condition.